Two-pore channels, TPCs, are ancient ion channels present in the cells of both animals and plants. In animals, including humans, these ion channels play important roles in biological activities in various tissues, such as in the brain and nervous system. All land plant species contain TPC genes. In many higher vascular plants such as Arabidopsis thaliana, Arabidopsis, and Oryza sativa, rice, a single TPC gene is involved in the activity of slow vacuolar SV channels, voltage-dependent cation channels, along with long-distance signaling, defense. However, very little is known about the function of TPC proteins in non-flowering mosses and liverworts, some of the oldest organisms on Earth. In a recent study, a team of researchers led by Professor Kazuyuki Kuchitsu from Tokyo University of Science, Japan, collaborated with researchers from Maria Curie Skłodowska University, Poland, to explore the evolutionary and physiological significance of two-pore channels in the non-flowering bryophyte. Their widely recognized and appreciated article, which discusses this study in detail, was first published online in December 2021 and subsequently in print in the February issue in Plant and Cell Physiology. The article has also been chosen as an editor's choice and research highlight article for the journal, which has published a commentary. M. polymorpha, or common liverwort, grows as thin, flat green sheets on moist soil or rock, and is an extant descended from one of the earliest plants to colonize land. M. polymorpha is a simple model organism that has been used to analyze the common characteristics of land plants. We realized that the genome of M. polymorpha has 3 TPC homologs, MPTPC1, 2, and 3, belonging to 2 distinctive groups, type 1 and type 2 TPC genes. We aim to know what these 2 subgroups of TPC proteins do in M. polymorpha, Professor Kuchitsu explains. To do so, the researchers first performed a phylogenetic analysis of the TPC genes in the green plant lineage. Then they characterized the 3 TPC proteins, MPTPC1 from the type 1 TPC gene and MPTPC2 and MPTPC3 from the type 2 TPC gene. Tagging these proteins with a fluorescent marker, they studied their localization in M. polymorpha cells. The results of the phylogenetic analyses provided some intriguing insights into the evolutionary history of M. polymorpha. Unlike the type 1 TPC gene, which is well conserved in all land plants, type 2 TPCs were found in algal species. This suggested that although the type 2 TPCs emerged before plants colonized the land, they failed to make their way into the genome of higher vascular plants and hornworts, Professor Kuchitsu tells us. The researchers also found that the 3 TPC proteins were primarily localized at the vacuolar membrane of M. polymorpha. The mutant that lacked a functional TPC1 gene showed no SV channel activity. But mutants that lacked either functional TPC2, TPC3, or both, exhibited usual SV channel activity. Molecules such as phosphatidylinositol 3, 5-bisphosphate and nicotinic acid adenine dinucleotide phosphate, that activate the TPCs of mammalian cells, failed to affect the ion channel activity in isolated vacuoles of the mutant plants. Professor Kuchitsu surmises, these observations, when tied down together, indicated that the type 1 TPCs, which are ubiquitous in all land plant species, are responsible for SV channels in their vacuolar membrane, but the type 2 TPCs likely encode ion channels that are different from the SV channel Na. With their eye on future research, they also aim to use insights from the evolutionary history of plants for improving plant growth and defense mechanisms against biotic and abiotic stresses. This could benefit industries like agriculture, among others. The funding for this research was obtained through a grant from Japanese Society for the Promotion of Science and the National Science Center, Poland. Make a difference. Sponsored opportunity story source. Materials provided by Tokyo University of Science. Note: content may be edited for style and length. Journal reference: Kenji Hashimoto, Mateusz Koselski, Shoko Tsuboyama, Helena Jubinska, Kazimierz Trebich, Kazuyuki Kuchitsu. Functional analyses of the two distinctive types of two-pore channels and the slow vacuolar channel in Marchantia polymorpha. Plant and Cell Physiology, 2022. 63, 2, 163 doi, 10. 1093, PCP, PCAB 176. Cite this page. Tokyo University of Science. Lighting the Tunnel of Plant Evolution. Scientists explore importance of two-pore channels in plants. Science Daily. https://ift. tt. 70h8xpj. Accessed May 2, 2022. Thanks for watching. Please subscribe my channel for more videos.